Okay, so that's turning an enzyme on. We also need to be able to turn our enzymes off, and that's called enzyme inhibition. Um, so enzyme inhibition, there's two ways. There's competitive inhibition and non-competitive inhibition. Competitive inhibition is if that cofactor binds to the active site. So like in this example, the cofactor bonded and turned the enzyme on. You could also have a cofactor bind and change the active site shape so the substrate could not bind and effectively turn the enzyme off. So that's if the cofactor binds to the active site. Non-competitive inhibition is if the cofactor binds to an allosteric site, which is just a different part of the enzyme that's not the active site. So let me show you what I mean. Here's an example of non-competitive, meaning the cofactor does not bind to the active site. So let's take a look at this example. We have substrate A binding to enzyme 1. Here is the active site. Here is the allosteric site. Remember, the active site is where the substrate binds. So A binds with E1, and that kicks off it's the first part of a metabolic pathway, which leads to F. Okay, now, once we have enough Fs, we want to turn this process off because we have enough. We don't want to waste using any more materials if we have plenty of F. So F, the end product, actually acts as the inhibitor or the cofactor for the pathway. So the end product binds to the allosteric site of the enzyme and changes the active site. So here, right, the active site was this crescent, right? Now it's this triangle shape. And so the end product, which is the inhibitor, binds to the allosteric site, changes the active site shape, prevents A from binding, and turns off this whole pathway. So this is a really cool example of how our bodies can turn enzymes on or off without needing it to produce anything extra. The end product is the off signal to the enzyme, because once enzyme 1 is turned off, none of these other reactions can occur, right? Because they all depend upon one another. So that's called feedback inhibition. I'm going to show you a video, a video kind of demonstrating what I just talked about.
Many of the enzyme-catalyzed reactions that occur in a cell, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, the product of one reaction becomes the substrate for the next reaction. If the end product of a pathway, such as an amino acid, becomes available in the environment, it is unnecessary and wasteful for the cells to continue to produce the product. Cells, therefore, have the ability to shut down a pathway when it is not needed. In feedback inhibition, the end product of the pathway reacts with the first enzyme that is unique to the pathway. The reaction occurs at a site on the enzyme that is different from the active site, called the allosteric site. When the product binds to the allosteric site, the enzyme undergoes a conformational change and can no longer react with its substrate. There is no substrate for subsequent steps in the pathway, and the final product is no longer synthesized.
So, say the end product, right, turns this whole thing off. And then our body's like, well, actually, wait, I need more of this end product, and I want to turn this system back on. All that needs to happen is that this gets released from the allosteric site. The active site will go back to normal, and the whole process will be turned back on. Now, sometimes these inhibitors can actually be bad because, like I said in the video here, this process can get turned back on, right? Like if this inhibitor unbinds here, then the whole process can turn back on and it's fine. But some inhibitors turn the enzyme off permanently. And there are several poisons that do that. One of them is cyanide. Cyanide turns the enzyme off that's needed to make ATP, right? And we learn ATP is super important, so if we can't make it, we die. Sarin turns the enzyme off that's important in the signaling process between your nerves and your muscles, so it paralyzes you. And warfarin turns the enzyme off responsible for blood clotting, and is actually a medication that's given to people to thin out their blood. It's a blood thinner. Okay, so let's recap enzymes. So if I showed you this biochemical pathway, if I showed you this enzyme picture, and I wanted you to label all of the letters, right, let's see what you come up with. So we have this circle here, binds to this particular part, and after that it turns the circle into a square. The square then binds to this particular part of whatever this is. After that, it changes it to this hexagon, which then can actually come down here, bind to a different part, okay, and that is called something. Alright, so what is A? A is the substrate. Good. And that particular part that the substrate binds with is called the active site. Great. And what are these blue things that we're talking about? Well, we're talking about enzymes. And what's the end result here called? Product. Good. Now, that product can come down here and bond to this other part of the enzyme called the allosteric site. Good. And when that happens, what is this process called? What is this arrow referring to? Good. Feedback inhibition. Okay, that is enzymes.